Hello, everyone, and welcome to the webinar. I'm Kate Osterly, and I'll be your host today. Thank you so much for joining us for today's live event. We'll be discussing a computational approach for locus-specific databases for rare diseases. We have a lot of great information to share with you, so I'm going to get right to housekeeping and our introductions. As you're watching today's event, please feel free to submit your questions in the Go To drop-down window, and if time allows, we'll get to those at the end of the presentations. This webinar is being recorded and it will be emailed to you later today. I encourage you to keep an eye out for that email and share that with your contacts and colleagues who find this information valuable. Now I have the pleasure of introducing today's speakers. I'd like to welcome Dr. Mark Kiel. He's the Chief Scientific Officer at Genomenon, where he oversees the company's scientific direction and product development. Mark received his MD PhD in clinical pathology at the University of Michigan, and he founded Genomenon to address the challenge of connecting pharma researchers with evidence in the literature to help diagnose and treat patients with rare genetic diseases and cancer. We're pleased to also welcome Dr. Gus Khursigara, the Vice President of Medical Affairs and Clinical Strategy at Inozyme Pharma. Gus received his PhD in molecular neuroscience from the Weill Cornell Graduate School of Medical Sciences. He has almost 20 years of experience in the pharmaceutical industry, with his last 12 years being focused on bringing treatments to patients with rare and undertreated diseases. I'm looking forward to today's discussion. So Gus, would you like to get us started?
Thank you, Kate, and thank you all for attending. I appreciate the opportunity to give a perspective from an early stage company on the value of having a partnership with Genomenon and how they're going to help us with some of our objectives and our mission at Inozyme Pharma. For those that don't know Inozyme, it's sort of a classic startup. It's 5 years old. We did an IPO not too long ago. And we are interested, as you see, in the unmet medical need for diseases with abnormal mineralization. And of course, we have an asset that we've been working on, an enzyme replacement therapy to address some abnormal mineralization. You know, when you go to the next slide, just to give you a bit of a background on a bit of the pathology, the clinical spectrum, and then what our objectives are. It just wants to set it up to how we're going to leverage our relationship with Genomenon. So to the left is a normal setup for mineralization. You see in the green in the bottom left there, ABCC6, it takes ATP and accelerates it from intracellular to extracellular. And then the ENPP1 will hydrolyze ATP to pyrophosphate or PPI and to AMP. AMP then is broken down to adenosine. Now, the value here is pyrophosphate or PPI is a very highly regulator for mineralization. So calcium and phosphate, the building blocks for mineralization, is necessary for bone. Too much of it, bones are brittle. Too little of it, bones are soft. Because it's ubiquitous, calcium and phosphate, as you can imagine, also circulate in the body. So PPI becomes also another important player to try to minimize calcification in the vessels and tissue. Adenosine also plays a role in arterial stiffness or arterial thickness. Now, to the right, if you have a deficiency in ENPP1, as exampled here in the middle, you are not creating or producing PPI, pyrophosphate, and you're not producing adenosine. The consequence from the physiologic perspective is calcification of arteries and thickness of the arterial wall. Thickness can lead to stiffness that can lead to stenosis. So how does this manifest in patients that do have a deficiency in ENPP1? What you'll see as we move to the next slide, this can present itself across a broad age range. And again, not so different from other inherited enzyme diseases. In the case for ENPP1 deficiency, what you see to the left is they can be identified as generalized arterial calcification of infancy, or GACI. And when born, they have tremendous extensive calcification of the arteries. You tend to see a lot of it in the aorta. It has a high mortality rate, as you see on the slide to the left, at 50% within the first 6 months of life. You can also see at the bottom some x-rays of a calcification around the heart and artery. You can see some stenosis from an echo. So even with survivors of GACI, the other 50%, they eventually move into where they develop skeletal complications, cardiovascular complications, neurological complications. There is also a group of patients that are identified with hypophosphatemic rickets that didn't have or did not present with GACI or identified with arterial calcification. They too also present with rickets due to the deficiency in ENPP1 enzyme. And again, as you can imagine, similar to other diseases that are skeletal in nature, you can see that as they get older, this has also an impact on mobility, on pain, and just overall risk of heart valve defect. So it does highlight, and this is why I bring it up for this discussion, it highlights it's heterogeneous. These patients present at any age. That means they touch a lot of specialists. And as it turns out, right now, this disease is considered very rare. So you can see the challenges that we are facing when we talk about trying to bring therapies to patients. On the next slide, very quickly, we do have a product, an enzyme replacement therapy with ENPP1, the extracellular component. And just quickly, you can see in an animal model, in red to the left is your ENPP1 and a knockout animal and that activity. And you can see the next 3 points as we increase the dose of the concentration, the ENPP activity is increased. And what you see to the right, the output, the PPI, as we highlighted earlier, the red there, the vehicle, there's no PPI in the knockout. And you can see that there's an increase of pyrophosphate when we introduce the enzyme. We've shown that we can recover failure to thrive. We can reduce vascular calcification. We see an increase in survival. So with that IND, we have put in and have received approval to move into our first in human study. And so the next slide is your typical multi-ascending dose safety study. We just announced on Monday the first patient has been dosed. So we are excited moving from a preclinical stage to a clinical stage. And this is typical, and not to get into details, but we'll have a cohort with a low dose. If the safety looks great and we look at the PD, then we'll move it to a second cohort with a second higher dose. The same principle, if it's safe, we'll move it to a third. And here we'll hopefully get safety as well as a dosing parameters for a drug to move into further trials. So the next slide is, even though we're doing in our first in human study as of this past week, there are still things that we are building within the community itself. So the activities typical for rare disease launch, right, is one of the big challenges is increasing awareness. Usually there's very few patients and there's very limited publications around because of the few patients. So how do you increase awareness for a disease that hasn't been well publicized? Number two, how do you accelerate diagnosis? As I highlighted in the slide, the age and the spectrum and the heterogeneity of the disease is so broad, it can be misdiagnosed. And therefore, how do you get physicians or clinicians to think about a rare condition and accelerate the diagnosis? Three, we try to understand the prognosis. So early in rare diseases, as some of you might know, few publications, the case studies, few clinician experiences. So trying to understand how the disease progresses longitudinally is usually a very difficult, or at least there's not a lot of information available early launch. We want to determine the prevalence and we want to determine treatment response, not to standard therapy as well as obviously to our potential drug of interest. So to the right, and why we started to consider working in partnership with Genomenon. Some of the ways to overcome and appreciate the approach that we have is, trying to get one source where the literature and the genetic information stand. We've talked to a lot of physicians that try to do genetic testing, they get the variant back, they're not sure where to look up, or it takes a lot of time and dedication to find literature that's scant. So we feel that this having a one source will actually be very helpful for clinicians and actually help increase awareness. And Mark will talk a little bit more specifics around that. We want to determine prevalence, trying to identify and expand the variants of significance will help us determine the prevalence of the disease based on, penetration, incidence, and prevalence. We want to accelerate genotype phenotype. Is there something prognostic about certain variants that will tell us the patient will accelerate or not accelerate to a poor outcome? We want to expand genetic confirmation. Many of these rare diseases, as you know, they don't have disease-specific symptoms. Take rickets, for example. Hypophosphatemic rickets can be due to a lot of reasons and pathology. It just lands on rickets. So we are trying to expand genetic confirmation to try to accelerate diagnosis, as well as expand our potential patients that could benefit from a treatment that we're trying to develop. And finally, prepare. Are there any patients that don't respond to our treatment? Is there a genetic underlying reason for that? And can we try to identify that sooner than later? So I think those are the sort of the approach we're taking to the left, and how we think the partnership and what Genomenon is doing to the right is how they're going to help work with us to really increase the awareness, the diagnosis, and hopefully better outcomes for patients with ENPP1 deficiency. So I'll leave it over to Mark to sort of talk through a little bit more about what they do specifically in terms of this challenge.
Thank you so much, Gus, for that introduction. Gus will be back when I'm done with my introduction to what Genomenon does and a bit of more detail on what Inozyme and Genomenon have done together with respect to ENPP1 deficiency. As Kate alluded to at the beginning of the broadcast, if you have any questions, I invite you to enter them into the chat, and we'll perhaps have time to go over those with Gus when he returns. So for my part, I want to share with you my experience in working with rare disease pharmaceutical companies like Inozyme and the work that we've been doing with Gus, lending a little bit of the insights that I've learned through that several years of effort. So I'd like to begin with some of the things that Gus talked about with specific relevance to ENPP1 deficiency by highlighting this as a problem across multiple rare diseases. In particular, rare disease diagnosis is challenged by a number of characteristics, and the early identification and diagnosis and treatment of these patients can dramatically improve outcomes, as Gus alluded to. So for rare disease, most rare diseases, 70 to 80%, have a genetic component, and most of those individuals with rare disease remain undiagnosed, whether throughout their life they remain undiagnosed, or there's a great delay in receiving a confirmed diagnosis. That's referred to clinically as a diagnostic odyssey. And statistics suggest that for rare disease patients on average, this diagnostic odyssey can last 4.8 years and result in seeing 7 or 8 specialists, as Gus suggested, for ENPP1 deficiency during that odyssey before they finally obtain the correct diagnosis. The other trend that we're seeing is that more and more patients are being sequenced with the advent of next generation sequencing technology and things like newborn screening expansion through sequencing, which is offering a great deal of promise for expediting these diagnoses for rare disease patients and improving outcome and mitigating this diagnostic odyssey. But the challenge there is that many of the genetic variants that result from sequencing are poorly characterized, and that puts a limit on the value that sequencing can provide. So to improve rare disease diagnosis, capturing existing knowledge about the disease genetics, as I'll speak to in subsequent slides here, and then ensuring optimal engagement with that information. So I'd like to say weaponizing that knowledge, not just keeping it precious and in a silo, but making sure that it gets out into the world on the front lines where the clinicians are seeing these patients to diagnose and treat them. And so when you put these two things together, we like to refer to the comprehensive organization of this evidence as a genomic landscape, which is to say every genetic variant and all of the supporting evidence necessary to adjudicate its meaningfulness clinically is aggregated and then integrated into clinical practice. And I'll speak to some of those specifics with the Inozyme Genomenon partnership that Gus has alluded to. So Genomenon's mission is to make genomic evidence actionable. We have a particular interest in rare disease because there's a great need in making this information actionable and getting it in the hands of clinicians. And in the service of better designing and better optimizing drug development and drug delivery. So that's our mission. And our goal, subserving that mission, is to curate the entire human genome. Doing so one gene or one disease or one set of diseases at a time. And we are a piece of the overall puzzle. We're a very significant piece, which is to say the empirical evidence in the published literature that's uniquely beneficial for clinicians to render their diagnoses and uniquely challenging to organize and annotate and adjudicate that evidence. And so there's a problem here in that extracting this meaningful evidence, especially for rare disease from the published literature, requires, or hitherto had required, manual extraction and navigation of the complexities of genetics and genomics and idiosyncratic clinical phenotypes and presentations. So what Genomenon has done is automated that process with sophisticated computational techniques. We take the published literature, identify the relevant aspects of that literature, the relevant articles, and extract the meaningful genomic concepts out of those articles, and then organize those into a network of genomic associations. What results from that process is the Mastermind Database of genomic associations, which comprises all diseases, all genes, and all genetic variants in those genes, as well as the attendant clinical and functional significance of those variants found in any one of the patients that had been published. So this is a clinical use tool that I'll speak to here in a moment, but it is comprehensive across the entirety of human disease, the full breadth of the human genome, as well as every variant, no matter how poorly described it is by the authors. The Mastermind capability allows us to comprehensively ingest and annotate and organize that evidence. We go further beyond just the computational organization of that evidence for maximal sensitivity, but we also have internal capabilities that allow us to curate that information for maximal specificity. So we have a Curation Engine capability that allows my team of curators to adjudicate that evidence fit for purpose, disease specific or drug specific, and present that evidence back into Mastermind, which is what Inozyme and Genomenon have been working toward in our partnership. And I'll show you the fruits of that work here in a moment. So that computational approach to annotating and aggregating the evidence and for maximal sensitivity, and the specific approach using our internal capability for maximal specificity by human curation allows us to produce a comprehensive genomic variant landscape. In this case for ENPP1, but we're able to do this across the whole human genome. So for every genetic variant in any rare disease or cancer or other disease, those results are interpreted to the clinical standards, annotated for clinical and functional significance, and all supported by the evidence. In addition to that, we are able to produce a comprehensive patient landscape, which is to say every published patient for any rare disease, in this case ENPP1 deficiency, all of which or all of whom are characterized by their demographics, the age and gender and ethnicity, the clinical phenotypes that attend their clinical presentation, the laboratory findings, the treatments that they received, and the outcomes of those treatments. So collectively, that information comprises a natural history study. And those in the audience may be aware that there's two basic flavors of natural history study. One looking back in aggregated data from the past and retrospective analyses, and the other looking forward, where you start with nothing and you start to accrete patients either in a cross-sectional study or in a longitudinal study looking forward. And so there's no reason for these two versions of natural history studies to be at odds. In fact, they're very complementary and very synergistic. And this table summarizes that synergy. Suffice it to say that the retrospective approach allows you to gain keen functional insight into the consequences of these genetic variants, as well as expanding your cohort size, particularly by capturing more of an ethnic representation of the disease. Whereas the prospective study, you can a priori define your own collected parameters, and you, by virtue of the longitudinal understanding of the patient's course, you can get deeper and more specific progression information about your patients. But again, both of those benefits blend together in a unique synergy that makes for a very robust and unique natural history study. And that's what we've done at Genomenon with Gus and Inozyme. So I just want to walk you through some of those data points and some of the details before I bring Gus back, and we'll have a conversation about the challenges attending rare disease drug development and delivery. So this is a summary of the source of the patients from the retrospective and the prospective natural history studies that I just alluded to for ENPP1 deficiency. So in blue there is resulting from our comprehensive patient landscape from the survey of the medical literature. And you can see a large number of patients, including 66 that were unique to the retrospective study. And then we actually had with our collaborators from the NIH and the University of Munster, we had 2 separate natural history studies that were prospective, and they contributed their unique numbers of patients to this consideration as well. And so I'm highlighting in this Venn diagram not how these differ, but how they blend together, and how there's a unique value in combining these 2 approaches. And you see the fruits of that unique value when you actually look at the variance that you uncover when you have a wide breadth of understanding from multiple different sources to aggregate this patient data. So particularly, I'm showing here the unique ENPP1 variants, obviously that are the cause of ENPP1 deficiency, GACI and ARHR2. And again, the Venn diagram is meant to say that there's complementarity here as opposed to pitting these 2 resources against, or multiple resources against each other. So we feel strongly that combining these approaches provides unique value and insight. I'm sorry that this is a bit detailed. I feel it's a bit obligatory, but this is the ENPP1 protein, the diagram on the bottom with the functional domains. And each of those dots or bars is a reflection of where those variants were found across all of the patients in our aggregated natural history study. And I'll emphasize, of course, that not only are these dots, they're patients, so they're reflective of patients who stand to benefit from early diagnosis and intervention. And all of these variants for each of these patients have been adjudicated according to clinical standards based on a comprehensive reflection of all of the evidence. So necessary and sufficient to ensure clinical appropriateness for diagnosis and treatment. And the last couple slides here before I bring Gus back on is to make this actionable. So as I say earlier, that's all well and good. We have a better understanding of the disease and how genetic variants cause disease, but Gus and I both are motivated to make this actionable. And the way that Genomenon is able to do that is by leveraging our reach into clinical laboratories in the service of connecting the patients who are diagnosed by the clinicians with the drug or the trial through the data, through the evidence. And for that purpose, we have the Mastermind clinical software tool that I alluded to earlier, used in hundreds of diagnostics labs across the globe. And this is what those clinicians see. Again, it's the evidence that they see. In the case of our partnership with Inozyme, in addition to seeing the evidence, they are able to see right away that the patient and the variant in their clinical workflow is appropriate for enrollment in the trial or prescribing of that drug if it's post-commercial. And so what I'm showcasing here is the clinician's eye view. When they have a GACI patient who's undergone sequencing, they can immediately see in our clinical software what that variant means, the fact that it causes disease, and the fact that there's a trial that that patient is amenable for enrollment in. So that's, as I say, a whirlwind tour of Genomenon's philosophy, the data composition that we have, and the way that we make value out of that data, and in particular, how the partnership with Inozyme and Genomenon is starting to bear fruit. So with that, I will bring Gus back on. Hey, Gus, and I'll also invite any of our audience members to weigh in with their own questions. I have a series of my own. Gus, you and I have been working together for some time now, so we can forget what we both think we know about each other. Let's just start with an open-ended question. What are some unique challenges for pharma companies focused on rare disease? We can go in any direction that you want to take that.
Sure. Look, I think I'll say this: the experience in the industry is getting greater and greater, right? There are more smaller companies that are trying to address rare diseases. So what used to be considered unique maybe 10 years ago, I think is becoming standard. And what is that? I think it all starts with just the definition. It's a rare disease. So when you get into a disease early stage, there are few patients. That means few clinicians. And that means what? There's not a lot of information and awareness that's available. And so the challenges of getting into a rare disease and launching a product in there is, how do you overcome that few patients, few clinicians? That means few publications. That means there's uncertainty where the patients are. How do you overcome that? And that's the general, I would say, Mark, a general response to what are some of the unique challenges. Now, we can break it down, if you wish, into different components, right? You know, if there are too few patients, how do you find patients? If they're too little experience, how do you broaden experience? And we can go down that road as well. But I think that's the underlying theme for any of these rare diseases: few patients, few clinical experience. How do you start to build it up?
So, you know, let's pull some of those threads. So one thing that you alluded to on your last slide, I think on the left, things that you at Inozyme are deeply engaged with now is the clinician education, which I feel like is a major challenge to mitigate the diagnostic odyssey and actually get the patient sequenced up front. So can you speak to some of the efforts at Inozyme for augmenting and accelerating clinician education in the service of diagnosing these patients earlier?
Yeah, no, it's the biggest challenge, and it's a challenge that I think it'll stay with us if we can develop the product, if we get approval, if we get payers to purchase, and we get into the commercial space. We will constantly be trying to educate physicians. Early on, the approach is there are a few physicians, usually smaller than a handful, that have published, that have interest, a very keen interest in some of these diseases. And it's to start there and learn from them. And it's a very good collaboration to start with those physicians, because you get to work together. As drug developers, you start to understand, or try to understand, you know, what are some of the endpoints that are important for a clinical development program. Clinicians will tell you what's practical and what's obvious. Now, from that perspective, you start to grow. You start to look for avenues of how do you target certain specialists. So how are these patients coming to the clinic in the first place? Who's looking at them? Who's diagnosing? How are they managed? In our case, and I think in many other rare diseases, there's multiple specialists that see this patient or can touch this patient. So we have to start to calculate, are we looking for people that are diagnosing? Are we looking for physicians that are managing? What are the key conferences that we go to? Where do we put our announcements? So it's not one specific path, Mark. I think in the end, we start there and we try to build the experience. Now, something like having a database like yours is interesting to us, because the first question that will come up is, how do I know it's really a Gacy or a Ricketts type or EMPP1? And as you know, we are now also looking to accelerate diagnosis by saying you should go and genetically test this patient profile. And when they go and test, and you probably know more than I do, clinicians will get that variant back and just not understand what that means. Right? Some physicians are okay. They see the phenotype, they see the genotype, and say, okay, I think this is EMPP1 deficiency. Others I'm not sure what that means. So having a data set like this early, even though we're first in human, is getting physicians to understand to genetically test. And then when we drive them here, as you highlighted, the literature is highlighted, patient advocacy could be highlighted, clinical trials could be highlighted, and it kind of gives a one source for that clinician to get more positive. So I think I rattled on a few little points there, Mark, but hopefully that sort of sets up the stage for some of the other questions we might get as well.
Yeah, just a little bit more detail about the gap between recognizing there's a challenge and pulling the trigger on a sequencing effort. And so I remember during my training, a decade or more ago now, there was uncertainty about the value of sequencing, because if you can't take action on it, or if there's an interpretation challenge, why bother, was the sentiment. I think it's now 2 decades actually that has been since training. Are you seeing a continued trepidation about casting a wide net and doing, say, exome sequencing or even large gene panel sequencing? Or are you seeing that barrier break down with the recognition that there's real value to come out of the sequencing, especially when you fully understand what the results may mean?
Yeah, and that's a great question. I think I'm still a little bit more toward your experience. My impression is, what I've seen is clinicians will diagnose to a treatment.
Yeah.
They won't necessarily, there are very few, a handful of physicians that will continue to try to keep pursuing what is the rationale for the pathology. But in general, and that's not a judgment, physicians treat to a treatment. Now, why would they look for GACI type of patients if the treatment is just going to be the same in terms of phosphate and calcitriol, et cetera? What we, when you have a company, a pharma company that's now looking for a therapeutic, it does create some motivation to further diagnose that patient, because you can then go down the sequence. Can they be recruited into a clinical trial? Would they be available for the therapy down the line? And again, I think that moves from diagnosis to a treatment. Now, in a rare disease, there are many diseases that don't have disease-specific symptoms. They all look alike. Again, I'll focus on rickets since we're on this topic. So that's where the genetics comes in. We will now go out and talk to physicians and talk about the prognosis and how this phenotype can look similar to other phenotypes, and genetics become important. Because one, we're recruiting, two, we also now understand some patients will not benefit from other treatments if they have our mutation. And so that's what I think inspires physicians, clinicians to move toward genetic and sequencing. And then as we hit upon is, are these physicians comfortable once they get the genetic sequence back? What do they do with it? But they have to be inspired.
So one other thing that you touched on in that last slide that you presented was, this is a rare disease, and that makes it challenging to estimate the number of patients that you can successfully treat. So I wonder if you can speak to some of your early challenges in estimating the prevalence, understanding the perhaps geographic distribution of these patients, and how that informed your strategy, thinking about developing the trials or thinking about post-commercial phases, even looking forward.
Yeah, Mark, I have to tell you that's part of the reason why we did a partnership with you, is that is exactly the information we need to try to figure out, to be honest. So what have we done? We've gone to different institutions and we've done some simple, just marketing. We've tried to understand through ICD-9 codes similar phenotypes, and maybe they're captured in there. I think some experts have tried to look at some of the genetics and using what we know from the number of mutations, looking at penetrance, and therefore then trying to size. What spurred us on is to try to understand from you, and what I think we're getting in our results is we've been able to identify more and more variants. And as we continue to identify more variants, either through the literature or unpublished, but yet physicians continuing to add to the database, we are starting to see a broader number of patients. So our approach has been a little bit manual. I would say it's probably not so successful, but it's just given us a perspective or a landmark to start with. But I would think in the next, and why we're doing this so early with you, right, is we want to encourage physicians to use and add their variants and their interest into the database so we can get a better prevalence. You know, as we move forward with regulatory agencies, and when we really look forward toward payers, we need to understand what this potential patient population looks like, because it sets up how we are going to strategically roll out, whether it's due to pricing, whether it's due to, as you say, areas. You know, there are certain countries we may or may not go to. So we're not there, Mark, but that's exactly why we're trying to spend the next 2-3 years building up this part of the database.
It's a particularly unique challenge in rare disease, and in conversations with multiple other groups, it's hard to, what is the right answer? I don't feel like there is a right answer. It's such a big question. So you have to rely on assumptions and estimates, and the quality and care with which you make those assumptions and apply them, I think that's really where you triangulate around the actual truth. And it starts with really understanding the disease, as you said. So you talked about the clinical heterogeneity and the sort of diagnostic challenges absent genetics, but even within genetics, what we have been working on is clarifying and minimizing that uncertainty within the genetics space. So particularly for GACI, you had that nice progression of GACI and rickets and osteomalacia across the different age groups. With specific relevance to GACI, can you speak to the phenotypic presentation and the timing and the acuity of the diagnosis for earlier intervention leading to improved outcomes?
Yeah, I'm happy to. So really, this ENPP1 deficiency was really initiated, or the gene was identified because of the GACI phenotype. And again, generalized arterial calcification in infants. So you can see even prenatally in ultrasounds some calcification that seems to be occurring in key arteries and around the heart, quite frankly. And that's what started this phenotype. Now, because of the calcification of the heart and artery, you get extensive calcification. There are many of these patients that actually die prematurely before birth. And then we see even after birth within days and weeks, and really I think I said up to 6 months, but really even earlier than that, you see that these patients will die from either cardiopulmonary issues. Now, even looking at the genotypes that's been attempted, are there particular mutations that are prognostic to patients that will die and patients that don't? And we haven't seen that so far. There's no predictive risk factor to this point that can be identified. And so that is one of the things that has always been of interest to many of the clinicians is can we predict that? And we can't so far. Now, we don't know why some survive and some don't. There's some speculation that because of the change in the metabolism, and there's a difference in phosphate to pyrophosphate ratio, that the body then starts to elevate FGF23. And what FGF23 does is it forces phosphate wasting, or patients start to pee out phosphate. And because of that, and since phosphate's part of that calcium building block, this might be part of the reason why we see some calcification resolve in some of these patients. Now, as they grow, what hasn't been well documented until we got involved is they start to develop rickets, because guess what? They're wasting phosphate and now they're undermineralizing. So it's a bit of a contrast between extensive calcification and undermineralization in these patients. So that phenotype early on for GACI is quite obvious and overt. That hasn't been an issue. Understanding why some patients survive and some don't has been a challenge. Once they survive, it looks like they all move into that hypophosphatemic ricket phenotype. And I will say, whether it's FGF23, whether it's a FEX mutation, whether it's a ENPP1 mutation, the phenotype looks very similar. And that's where the genetic confirmation becomes more critical to us, both from a diagnostic perspective, but as we try to identify patients and recruit patients for any future trials. So hopefully that gave a little bit of an answer there.
To build on that, you mentioned FEX, and we actually have an audience question that asks more generally, how many diseases have more than one genetic anomaly. And so you talk about effects with respect to the sort of clinical overlap in phenotypes.
Yeah.
On I think your second or third slide, you mentioned ABCC6 being part of this ENPP1 pathway. And so where I know that ABCC6 mutations do not often cause GACI, they can you speak to the genetic complexity of this constellation of genetic defects and how the value of the data that we produce can start to penetrate through that complexity?
Yeah, I'm going to go in reverse. That's what we need, right, Mike? We need this type of data to address that type of question. So it's a good question, especially since I hit on FGF23. So what we've seen, or some clinicians have told us, is they will see patients that come in and they diagnose them as XLH, and that's XLH is a hyperphosphatemic ricket due to FX mutation. When they go through genetic testing, in some cases, payers are asking for genetic confirmation that is truly FX mutation to be given treatment, burosumab. It turns out that they don't actually have FX mutation; they have an ENPP1 mutation, right? So now, and why is that? Because as I highlighted, ENPP1 deficiency elevates FGF23, FX mutation elevates FGF23. So it's not a surprise you get to see the same phenotype. What is even more interesting is in that pathway I showed, you're right, there's an ABCC6 that translocates intracellular, extracellular ATP. ENPP1 breaks down PPI at AMP. You have a CD73, which I didn't highlight, that breaks the AMP into adenosine. But there's also then ALP that breaks down PPI. So PPI can be, and adenosine can be managed by other enzymes. Are the mutations there? Is some deficiencies there? And there are some patients we've seen, and I say a small amount in this case, that have both an ENPP1 and an ABCC6 mutation. Just to what's the word I'm looking for? Just from a personal conversation and no other evidence we've seen, there are some FX mutation that look like they also carry an ENPP1 mutation. So this is where I think, this type of database will be helpful. It's not from a competition, and this is what's so interesting, right? It's not a competition, to try to understand what's causing the phenotype. I think what is value here is dissecting out what is the dominant gene or the dominant loss of function that's leading to the phenotype, because any company that's working to replace some of these enzymes, you want value and response from your drug. You don't want to introduce a therapeutic if it's not addressing your gene. And so, I don't have a good answer for that. What I'd say is I think we recognize there's some overlap. Does that make a difference? Is ENPP1 more or less important than FX mutation? We don't know.
Yep. I like the way you frame that. Is in addition to not wanting to needlessly treat a patient with a treatment that will be inefficacious, if you're in the context of a trial, you want to be sure that you optimize the chance for successful outcomes for that trial. And so clearly understanding what the genetics are underpinning that individual's pathogenesis, and obviously understanding your drug mechanism, making sure that there's a match there. I can actually address, from my experience as a molecular diagnostician, this is true of cancer. Cancer in the 70s, 60s, 70s, war on cancer was like this single enemy. And what we learned in that war was there's just, dozens or hundreds of different enemies with each of their own idiosyncrasies. And so there was, even during my training in hematopathology, there was a parsing of the diseases that looked the same, but the underlying genetics were different. This is also true in neurodegenerative disease with ALS, and it's particularly important as precision medicine comes to the fore, whether it's enzyme replacement therapy or antisense oligonucleotide technology. It sort of drives home the point that really clarifying the aboriginal cause of these diseases at the genetic level is going to be really important in most of these circumstances. So another question that I had while you're speaking is, as I alluded to earlier, the clinical reach of Mastermind is across thousands of diagnostics labs across the globe. Can you tell me what it means for you to be able to find one of these rare disease patients. I imagine that there's a bit of euphoria when you're able actually to identify one of these patients because you feel that you can have a positive impact on their lives.
So yeah, it goes both ways. One is it's a positive impact. There are sometimes we find clinicians that are treating patients or families of patients that just desperately want to understand, why are they not responding to current therapy? Physicians also, physicians want to get the right therapy as well. So sometimes just identifying why a therapy is not working because they have a mutation in the ENPP1 is very joyful. I'll say that first. We get excited, as anybody in a rare disease knows, every patient does count. It's not a saying, but it's just the way you have to live, especially when you're in a stage where we are, for example, at Inozyme, because we are still trying to learn about the progression of the disease. We are still trying to learn how they were identified. So every time we see a patient, it is euphoric in a way, to your point, both from just a family perspective as well as from a scientific perspective, and then maybe at some point from a research perspective from a clinical development. So that is why, and I didn't hit upon it on one of your questions earlier, as we're going to physicians, the other thing we are trying to do is go to diagnostic labs, right? So do they include a genetic panel that includes ENPP1? And listen, some did and some didn't. And as we continue to build evidence and the value of why they should offer it, we're seeing more and more labs pick it up. I don't think that's unique to us, but I think as labs get understanding on value, they're going to add more to the genetic panels. And so again, this is where I think genes will play a role. We may start to, our hope is you'd like to see that anybody with hypophosphatemic rickets, when they're getting a genetic screen, will include all the genes that we've been highlighting and then some. And you're not going in to talk about one disease, but you're going in to talk about a panel and what is the cause or what's the direct cause of that. And so that's what our hope is working with diagnostic labs to include ENPP1. And then, Mark, that's one of the things that we think through is as these diagnostic labs are identifying genetic variants, how we're going to make sure they get captured and pulled into Mastermind. So we continue to grow our own understanding of genetics within our own diseases.
Pursuant to a question, again, from an audience member, but also harkening back to some of my slides, I wonder if, I'll remind what we did here. We merged the retrospective with the prospective studies, two different prospective studies, and produced in aggregate a larger and more insightful data set. The asker asks about how these clinical complexities and genetic complexities could potentially confound that merger. And I'll just emphasize that that's exactly the painstaking approach that we took, was to be sure that we did not confound the analysis. We understood very clearly what the genetics was for each patient, and very clearly what the clinical features were, not just what the diagnosis was, but what the attendant phenotypes were, the age of onset, the different diagnostic modalities, clinical lab values, etc. Data is messy, especially clinical data, and you have to start from the cleanest, most comprehensive data set that you can. And then when you marry the retrospective with the prospective, which were treated similarly, right, similarly detailed, can you speak to the value of synergizing those two data sets, particularly for a rare disease?
Yeah, and so just to be clear, the prospective that we're highlighting, our terminology here isn't from a classic prospective design study and we went ahead and did a study. The prospectively here is at the NIH, they prospectively collect data over time because again, they have a physician there that is very interested in this disease and how it progresses. So from that perspective, you can call it prospective, but really this was a chart review. Now, it was really good from both the Munster from Europe as well as the NIH in the US. Because it's a rare disease, these two work together. So the data that was collected was agreed upon in terms of, let's call it a similar protocol, if you will. So it's a chart review, just to be clear, and they both agreed to capture the same information because they wanted to aggregate and increase the patient numbers. And that became very helpful. for obvious reasons. And I think that might address some of the questions of where there's some confounders in there. As Mark highlighted, we had tracked the same data set, the same clinical outcomes, and so we understood much better how the phenotype looked in these patients and at the age of that phenotype for these mutations.
I'll speak to my own research experience in my PhD phase. You have to fight the tendency to change your experimental modality when you've learned something from the first iteration, because then you can't have compatible data. And so there's the necessity to be sure that you lock down the right parameters and the right collection techniques so that there is compatibility across data sets, which is hard when you have two sites. It's hard when you have clinical heterogeneity or genetic complexity. It's especially hard when you're trying to make inferences across multiple related diseases, because you've got to coordinate those different groups. And so one of the ways that you and our other clients have benefited is understanding what those outcomes and those parameters may look like a priori. So not only is there synergy between the retrospective and the prospective, but there's benefit to doing them in series, right? Yeah. So we worked on these in parallel and came together, but doing that in series, looking at the retrospective to inform the way that you set up your prospective, particularly if there's complexities and nuances that you need to disentangle to get a real good handle on, say, the genotype phenotype correlations that you're talking about.
Yeah, and so we didn't touch upon that. So while we're looking at the genetics and focused here, what collaborating with those two investigators and their sites and their information is trying to help us determine what clinical endpoints should we be looking at and determining in our clinical trial. Because again, and you see this in many other rare instances, at least in my experience, usually when you start off in a rare disease, it's the overt symptom that catches everyone's attention.
Yeah.
But as you start to break down whether it's the genetics or the protein, as you get to inspect, you start to see other patterns develop when you get more and more patients involved. And so what that retrospective and that chart review is doing for us is, are there other patterns that weren't so overt? But guess what? Multiple patients are showing the same thing. And can you explain it with our physiology? Maybe that should be an endpoint. And so that's part of it as well, on why these prospective, retrospective sort of progression natural history studies are critical, even again, as I say, at our stage of this point.
Yeah, in this context and in others, I've heard that you're very right. Sometimes on paper, the clinical presentation or phenotype that seems like it's the most important or the most appropriate for an endpoint is actually not the one that would improve quality of life if corrected, or that should actually be tracked for, final outcomes, which is to say survivability. It's sometimes it's diarrhea, it's not seizures, as an example. Yeah. And so really appreciating that beforehand, before you design your prospective endpoints and data parameters is important. I'm looking at the clock here. One last question, and it's forward looking and somewhat optimistic, but with the benefit of this aggregated comprehensive data set, if you do see a heterogeneity in response, if you do happen to have differential pathogenesis, even slight, because of this mutation or that mutation, and that may have bearing on the success of your intervention, I wonder if you could speak to how the data would be used after the trial, when the data has emerged, and looking at and reflecting on what that data may mean.
Yeah, so it's a really good and scary question in that sense, right? We like to think that when we offer a therapy to patients, in general, all patients will respond, and they all will respond with equal magnitude, and that's not usually the case. So there are many studies that show that either some patients don't respond, like very marginal, and you'd call them a non-responder, or they don't hit the target. What the genetics, I think, will do is try to figure out if it's the way our drug is designed and targeting, or is there another biology behind it on why they're not seeing what they're seeing. So for example, we've seen this in the old days with just general drugs being, metabolized much quicker, for example, through liver, right? And we come back with some of the isozymes of the liver through metabolism. And so when people back in the day or patients would have no response, sometimes they were just metabolizing the drug too quick. And right, you'd have to adjust, right? And then we started to look for some of these isoforms that to understand if they're going to respond or not. I look at it the same way, just to be clear. I don't look at that we're only going to take the top echelon and we're not going to provide some sort of support. But it helps us, one, manage expectations. You never want to tell a patient this is going to work 100% at 100%. You've got to set expectations, and this will help us do that. And number two, are there particular confounders or particular reasons why we're not seeing as great a response in some patients versus the other? Is there something we can do about that? And so that's how I look at it. I know sometimes I've worked with others where they get worried that we're going to cut down the patient population from here to here, but that's not what this is about, right? This is about how do you optimize what you have. And that's how the way I think we've been looking at it from a genetics perspective and response to treatment perspective.
Well, Kate, I'll call you back to the stage. Gus, that was awesome. As always, I always enjoy our conversations.
Yeah, that was good. Thank you. I appreciate the time. Yeah.
Thank you, audience. I will be signing off and pass it over to Kate.
Yes, thank you both so much. Yes, thank you, Gus. And thank you everyone for watching today. If you have any questions for our teams, feel free to contact us at the websites you see on your screen. Again, I'll be sending the recording to this webinar to today. And thank you again for joining us. Have a great day.